students, in this video we are going to discuss a very interesting topic of renal regulation of acid-base balance, right? And also renal tubular acidosis, various types of renal tubular acidosis, right? It's a very interesting mechanism, right? If you understand this mechanism clearly, it will also help you in better understanding of other disorders, right? So let's first of all try to understand how exactly our kidneys regulate our acid-base balance, right? As per regulation of acid-base balance is concerned, kidneys have basically two jobs, right? Their first job is preservation of bicarbonate, right? This bicarbonate is very precious for the body. So all this bicarbonate has to be reabsorbed, right? Their other job is excretion of H plus ion, right? In all this protein metabolism, right? Lot of H plus ion is being continuously generated inside the body. This H plus ion need to be excreted out, right? So their first job is preservation of bicarbonate ion, and their next job is excretion of the H plus ion, right? As per preservation of bicarbonate ion is concerned, right, we will just do some simple calculations, right? How much is your GFR? Your GFR is 180 liters per day, right? So how much is plasma bicarbonate level? It is 24 milliequivalents per day. Plasma bicarbonate, right? So with these two things we can calculate how many milliequivalents are excreted out per day, right? So excretion of bicarbonate per day, right? Excretion of bicarbonate per day will be how much? 180 into 24, right? This will give us 4320 milliequivalent per day. This much bicarbonate is actually filtered out per day, right? But how much is actually excreted out, right? The bicarbonate which is actually excreted out is just 2 milliequivalent per day, right? So out of 4320 which is filtered out, right, excreted out is just 2 milliequivalent per day, right? So what does this mean? That 99.9% of bicarbonate is reabsorbed. So what is the reabsorption rate in bicarbonate? 99.9% is reabsorbed, right? And what is the major site of reabsorption of bicarbonate in our nephron? It is our PCT. PCT is the major site for reabsorption of bicarbonate, right? Now how exactly PCT reabsorbs bicarbonate? The mechanism we will see, right? Now let's enlarge one cell of PCT, right? On this side of PCT we have got our main hero, our sodium potassium pump, right? So if sodium is being pumped down, that means sodium inside will be less, right? Apart from this, we have got H2O plus water combining over here, right? H2O plus CO2, right? They will form H2CO3, right? Then it will form H plus and bicarbonate, right? Now this H plus wants to go this side, right? But for going to this side, it will take the help of sodium hydrogen exchanger. Since the sodium is already less inside, and inside the tubule, the sodium is at the same level of the plasma. So there is a gradient of sodium from this side to this side, right? So we will have an exchanger. This exchanger will utilize the gradient of sodium. Sodium can easily go from this side to this side because sodium is already less over here. So the sodium will come from this side to this side, and H plus will go from this side to this side. So this is sodium hydrogen exchanger. It's very important, right? We also call it NHE3, right? We will understand its medical importance later, but at this point you need to know that since sodium potassium ATPase pump has pumped the sodium outside, this has reduced the sodium inside the cell, and this gradient is being used by this sodium hydrogen exchanger. So this H plus will come out, right? Inside we have got all the bicarbonate which is getting filtered out, right? So we will have a lot of bicarbonate which is freely filtered. This bicarbonate will combine with this H plus ion to form H2CO3. This will further break down into H2O plus CO2. This H2 and CO2 will be reabsorbed, right? This bicarbonate will go to this side with the help of bicarbonate chloride exchanger, right? Bicarbonate will go out and chloride will be pumped in, right? There is also one thing called sodium bicarbonate co-transporter, right, which will pump your sodium and bicarbonate together, right, in the ratio of 3:1, right? 3 bicarbonate for every sodium which is pumped out, right? So as simple as that, right? The bicarbonate is not being directly reabsorbed, right? The bicarbonate is combining with H plus ion, getting converted into H2 and CO2. This H2CO2, again with the help of carbonic anhydrase, right? We have got cellular carbonic anhydrase and luminal carbonic anhydrase, right? This H2O plus CO2, again with the help of carbonic anhydrase, will break down into H plus and bicarbonate, and this bicarbonate will be reabsorbed. So we can say that this bicarbonate is reborn over here, right? It changes its shapes and gets reborn over here and reabsorbed. Even while you are seeing that this H plus is going out, right, this H plus is not exactly being excreted out of the body, right? This H plus is simply combined with bicarbonate and resulted in net reabsorption of bicarbonate, right? So bicarbonate is being reabsorbed, but there is no net excretion of H plus ion, right? Net excretion of H plus ion happens in our intercalated cells, right? Intercalated cells. We will draw intercalated cells over here, right? In intercalated cells, what we are going to see is that H2O will again combine with our CO2. Again H2CO3 will be formed with the help of carbonic anhydrase, right? Again this will break down into H plus and bicarbonate, right? Now this H plus can get excreted on this side with the help of two transporters, right? Either H plus ATPase, right? Either H plus ATPase or H plus K plus ATPase. It can get transported via this route also, right? The K plus will go in and H plus will be excreted out. This is called H plus K plus ATPase. Okay. Now this H plus which is being excreted out, it will combine with the help to our titrable acid HPO4 2 minus, right? Right? And then after combining with this H plus, what it will become? It will become H2PO4 minus and excreted out, right? As earlier discussed, kidneys has got two jobs. One is reabsorption of bicarbonate. Other job is excretion of the H plus ion. But this H plus ion gets combined with two forms after being excreted out. One is your titrable acid and other is your ammonia, right? Titrable acid we have written, and now we will discuss how exactly it combines with ammonia to form NH4 plus ion. the whole story of ammonia starts in your PCT actually, right? The whole story of ammonia starts in your PCT. We will just enlarge one cell of PCT on this side, right? Now how the whole story will unfold, right? It will start with glutamine, right? Now this glutamine will be acted upon by glutaminase, right? Which enzyme? Glutaminase. Right? This will break glutamine into NH4 plus ion and glutamate. Right? This glutamate will further break down into alpha ketoglutarate and one more NH4 plus ion. This alpha ketoglutarate will get further converted into succinate and this succinate will give us 2 bicarbonate ions. So every time one amino acid of glutamine undergo metabolism, what you get? You get 2 NH4 plus ion and 2 bicarbonate ion. So we can say one amino acid of glutamine breaks down into 2 NH4 plus ion and 2 bicarbonate ion, right? Now this NH4 plus is impermeable to the cell wall, right? It can't cross this membrane and enter inside the tubule, right? So this NH4 plus actually breaks down into NH3 and H plus, right? So this H plus will enter with the help of sodium hydrogen exchanger, the same sodium hydrogen exchanger which we are seeing over here. It will enter with the help of sodium hydrogen exchanger. So sodium will be reabsorbed, right? And hydrogen will be excreted out. This NH3 is freely permeable, right? So this NH3 can permeate into this. After crossing, this NH3 will again combine with this H plus and become NH4 plus, right? Now this NH4 plus has entered inside the tubule and we will think that now it will be excreted out into the urine, right? But the story of NH4 is not so simple, right? There are a lot more adventures involved in the story of NH4 plus. Let's see what it's going to do further. So this NH4 plus will cross the whole of loop of Henle, right? And then it will come over here, right? What we are having over here? We are having 1 sodium, 1 potassium, 2 chloride transporter. 1 sodium, 1 potassium and 2 chloride symporter, right? And what we are having over here? NH4 plus, right? NH4 plus. Now this NH4 plus ideally should not be able to cross through this co-transporter because it has got spot only for 3 atoms, 1 sodium, 1 potassium and 2 chloride. So this NH4 plus should not be able to cross through this ion channel, right? But it kind of impersonates. It becomes a impersonator, right? It impersonates which ion? It impersonates potassium ion. And how it is able to impersonate potassium ion? Because both NH4 plus and your K plus, both NH4 plus and the K plus have same hydration ionic radii. Their radius is similar and charge density is similar. This phenomena is called atomic mimicry. Right? Atomic mimicry. Right? Because radius is same and charge density is same. Same hydration radius, and charge density is also same. Right? Now, this NH4 plus is going to mimic our potassium ion and it will come outside over here, right? Now, this NH4 plus, what it's going to do now? It's going to break down into NH3 and H plus. It will form an equilibrium reaction. So both these form coexist, a kind of cyclical equilibrium reaction, right? And what this serves? This serves as a kind of buffer. Buffer of what? We will discuss. This NH3 is actually freely permeable, right? Freely permeable to the membrane, right? So this NH3 is going to enter inside the membrane, right? And what it will get? This NH3, let's write in blue, this NH3 will take the H plus ion from here, right? And it will become NH4 plus. Now this NH4 plus can't diffuse back into the interstitium because it is impermeable to the membrane, and this is called diffusion trapping. That means this NH3 can come inside, but after fusing with H plus ion it can't go back. This phenomenon is called diffusion trapping. Right? And one very important thing which we can see over here is that this thing is acting as buffer. How as a buffer? Suppose there is some disease, some metabolic disorder, and lot of H plus ion are coming from this side, lot of H plus ion. Now these H plus ion are going to combine with our NH3, right? If they combine with our NH3, it will become NH4 plus. So we will have less NH3 over here, right? If there's less NH3 over here, so this gradient of NH3 will increase. So you will have lot more NH3 coming inside the tubules from this buffer, right? So if you get lot more acid getting excreted out, it will combine with NH3, it will make it NH4 plus, right? Reduce the level of NH3 and the gradient will increase and lot of NH3 will come inside. So NH3 is a buffer just in case if there is excess acid secretion, right? This NH3 will come to the rescue, right? And which is more dominant, the NH3 or H2PO4, right? This NH3 reabsorption of H plus is more dominant in managing our acidosis, right? Means roughly 50 milliequivalents of acid is excreted out of the body, out of which 30 is absorbed by your NH4 and 20 is absorbed by your NH2, right? So total 50 milliequivalent is excreted out, right? And why we need to reabsorb? Why can't H plus be excreted out on its own, right? Because if we have got lot of H plus ion over here, these ion pumps will stop working, right? These ion pumps have got a limit. They can't pump if the gradient across this membrane becomes more than 1000, right? So what is the pH of our cell? 7.4, right? So if the gradient becomes more than 1000, right, that means if the urinary pH is 4.4, right, the urinary pH can't be less than 4.4, right? If urinary pH is 4.4, right, at this point the gradient will become more than 1000, right? And these ion pumps won't be able to pump through this. So all this free H plus ion need to be reabsorbed, need to be absorbed into this H2PO4 or NH3, right? You know the reason now? This pH 7 and pH 6, right? 7, pH 6, pH 5, right? So pH 6 has got 10 times more hydrogen ion than pH 7, and pH 5 has got 100 times, right? So it's a log of 10, 100, right? So pH 4.4 has got 1000 times more hydrogen ions compared to pH of 7.4. So at this concentration the ion pumps will shut down, so we can't afford lot of free H plus ion inside the tubule, so they gets reabsorbed, they gets absorbed into H2PO4 and NH3, right? Now since we have drawn all this, right, we can discuss our renal tubular acidosis, right? First of all, we will discuss the proximal renal tubular acidosis, or called renal tubular acidosis type 2, right? In proximal renal tubular acidosis, the NH3 is involved, NHE3. This transporter is involved, right? So in renal tubular acidosis, we also call it RTA2, right? Or proximal renal tubular acidosis. Now think of it. If this NHE3 is not working, that means this H plus ion now won't go inside the tubules and the sodium won't be reabsorbed, right? If sodium is not getting reabsorbed, of course there will be diuresis. But just think of it, if this H plus ion is not going outside, it won't combine with the bicarbonate, right? If it doesn't combine with the bicarbonate, it won't get converted to H2CO3 and it won't be reabsorbed. So this bicarbonate won't be reabsorbed if this H plus is not coming out, right? If this bicarbonate is not reabsorbed, it will be simply excreted out in the urine, right? So in proximal renal tubular acidosis, you will have massive bicarbonate wasting, right? What you will have? Massive bicarbonate wasting. Right? So in which diseases you can have your renal tubular acidosis type 2, right? You can have it in multiple myeloma, right? So in multiple myeloma we can have renal tubular acidosis type 2. Right? And of course also in acetazolamide therapy, right? In acetazolamide therapy, which we give for mountain sickness, right? We have discussed in previous class, there will be also excretion of bicarbonate, and we need that, right, to get rid of respiratory alkalosis, right? Acetazolamide therapy, and also in tenofovir therapy. And also there is one drug called tenofovir. In that also you will get excretion of bicarbonate, right? Now this is about renal tubular acidosis type 2, your proximal renal tubular acidosis, multiple myeloma, acetazolamide and tenofovir. Since the H plus is not getting excreted out, there will be massive bicarbonate wasting, right? Now what happens in renal tubular acidosis type 1, right? That is your distal renal tubular acidosis. Okay, now we will write over here RTA1. Right, this RTA1 or distal renal tubular acidosis what will happen in this case? Your H plus ATPase will be defective, right? So H plus is not getting excreted out. Either H plus ATPase or H plus K plus ATPase, right? Or some problem with your carbonic anhydrase enzyme, right? Or some problem with this bicarbonate. It will be excreted out now. It will be excreted out through bicarbonate chlorine exchanger, right? So there can be some problem with the bicarbonate chlorine exchanger. So any problem with these, right? Any problem either with H plus ATPase or H plus K plus ATPase or carbonic anhydrase or bicarbonate chlorine exchanger over here can lead to renal tubular acidosis type 1. Now apply your logic and think what can happen, right? If this H plus is not getting excreted out, this K plus won't be reabsorbed. If this K plus is not reabsorbed, what it can lead to, right? It can lead to the whole K plus will stay over here, it will be excreted out. So it can lead to hypokalemia, right? So you will have hypokalemia in case of renal tubular acidosis type 1. Right? Apart from this, if this H plus is not getting excreted out, right, it will stay inside the body, right? So this can lead to what? This can lead to acidosis, right? So if this H plus is staying inside the body, you will have acidosis, right? If there is lot of acid inside the body, right, what it can lead to? Lot of acid inside the body. This excess H plus, right, will go and combine with your bones, right? This bone also act as a kind of buffer, right? It has got salts. So this H plus will go and combine with your bones. H plus CaCO3, right? It will make HCO3 minus plus Ca plus, right? So it will neutralize the excess acid. Your bones are going to neutralize this excess acid, but in turn it has to release out calcium, right? This calcium will be released out, right? And what else? Since the body is having lot of acidosis, so what body will try to do? Body will try to reabsorb lot of citrate, right? Because citrate can give you 4 molecules of bicarbonate. So body will try to reabsorb lot of citrate, right? If lot of citrate is reabsorbed inside the body, now all this excess calcium which was released from the bones will get filtered out and come over here, right? So you got lot of calcium inside the filtrate, but lot of citrate has been reabsorbed. Now calcium does not have anything to bind onto, right? Okay, bones have released calcium, the calcium has filtered out and reach over here, right? But the citrate has been reabsorbed, right? Since the citrate has been reabsorbed, there is a lot of free calcium, right? Free calcium will finally form your renal stones, right? Okay, so apart from this, the citrate will be reabsorbed, reabsorption of citrate. Since the citrate is reabsorbed, you will have free calcium, and this free calcium will lead to renal stones, right? Finally, you will get renal stones over here. right? Then there is something called type 3 renal tubular acidosis, right? Type 3. Now type 3 renal tubular acidosis is actually combination of type 1 and type 2, right? Then there is something called type 4 renal tubular acidosis, right? Type 4 renal tubular acidosis. Now, what goes wrong in type 4 renal tubular acidosis? Type 4 renal tubular acidosis is characterized by aldosterone deficiency. The aldosterone becomes deficient, right? Okay. Now just think of it. What aldosterone does? Aldosterone is going to reabsorb your sodium and excrete out potassium, right? So what will happen if aldosterone becomes deficient? Enough sodium won't be reabsorbed and enough potassium won't be excreted out, right? So in this case, enough potassium won't be excreted out and enough sodium won't be reabsorbed. If enough potassium is not excreted out, right? So enough potassium is not entering these intercalated cells, right? If enough potassium is not entering these intercalated cells, enough H plus is not coming out. If enough H plus is not coming out, what it will lead to? Acidosis, right? So rise in aldosterone leads to alkalosis, but fall in aldosterone leads to your acidosis, right? So less aldosterone means acidosis. So type 4 RTA is characterized by aldosterone deficiency, which ultimately leads to your acidosis, right? But what is the main difference over here, right? That in type 4 you will have hyperkalemia because enough potassium is not being excreted out, right? But in this thing, type 1, you will have hypokalemia. Both are acidosis, right? Type 1, you will have hypokalemia. Let's see. Now this aldosterone deficiency, right? What we said, it will lead to hyperkalemia, right? Hyperkalemia. And this hyperkalemia is going to inhibit your glutaminase, right? If the glutaminase is inhibited, right, NH4 plus won't be produced in adequate amount, so it is going to impede your H plus ion secretion, right? Because glutaminase is being inhibited, and hypokalemia is going to stimulate your glutaminase. It's a very important concept, right? Stimulate. Hypokalemia will stimulate. It's a very important concept that rise in aldosterone will lead to alkalosis, but fall in aldosterone will lead to acidosis. So in type 4 renal tubular acidosis, we have got aldosterone deficiency, which leads to acidosis, but which also leads to hyperkalemia. This hyperkalemia is going to inhibit your glutaminase level, right? And impede the excretion of acid ion, right? So we discussed all the 4 types of renal tubular acidosis, right? So it's all these mechanisms are quite interesting, right? So we'll just try to sum it up, whatever we studied, right? So this thing, what kidney has to do for regulation of our acid-base balance? Kidneys have to preserve the bicarbonate and kidney have to excrete out our H plus ion. For preserving the bicarbonate, the main job is of PCT, right? We also calculated the amount which is excreted out, roughly 4320 milliequivalent per day, right? It's filtered, but excretion is just 2 milliequivalent, right? So 99.9% of bicarbonate is actually reabsorbed. How it is reabsorbed? Our CO2 and water combines, form H2CO3, which further forms H plus and HCO3 minus, right? Now this H plus will be excreted out through sodium hydrogen exchanger called NHE3, right? This H plus will combine with our bicarbonate to form H2CO3, which will further break down into H2O and CO2, which will be reabsorbed. So this, by indirect method, this whole bicarbonate is reabsorbed, but there is no net secretion of H plus ion. Net secretion of H plus ion happens in our intercalated cells, right? The H2O combines with CO2, H2CO3. H plus can get excreted out either through H plus ATPase or H plus K plus ATPase, right? It will combine with our titrable acid HPO4 2- to H2PO4 minus. It can also combine with ammonia, but the story of ammonia starts in our PCT, right? This glutamine, under the effect of glutaminase, will break down into glutamate and NH4 plus ion. This glutamate will further break down into alpha ketoglutarate and another molecule of NH4 plus ion. This alpha ketoglutarate will break down into succinate, and this succinate will break down into 2-bicarbonate. So every single molecule of glutamine gives us 2 molecules of NH4 plus and 2 molecules of bicarbonate. This NH4 plus can't cross the cell, so it will break down into NH3 and H plus, right? And this NH3 can freely diffuse, and H plus will go inside with the help of sodium hydrogen exchanger. NH3 and H plus will combine to form NH4 plus. It will reach all the way up to this point. Then it is going to use our 1 sodium 1 potassium 2 chloride, cross to this side with the help of atomic mimicry, because it has got same charge density and same hydration ion radius. This NH4 plus will break down over here into NH3 and H plus, right? It will form an equilibrium reaction, right? This NH3 can freely diffuse into your collecting duct. Here this NH3 will combine with H plus and become NH4 plus. This NH4 plus can't go back, and this phenomena is called diffusion trapping, right? And more H plus, just in case if more H plus comes out, it will combine with more NH3, right? The level of NH3 on this side will go down and more NH3 will come out. So this acts as a buffer to handle the excess acid coming from intercalated cells, right? Now, since we have discussed all this, we can discuss our renal tubular acidosis. So in type 2 renal tubular acidosis, called proximal renal tubular acidosis, NHE3 will become defective, right? NHE3 becomes defective, and if this H plus is not going out, this bicarbonate won't get reabsorbed and it will be excreted out. So there will be massive bicarbonate wasting, which is classically seen in multiple myeloma. It is also seen in some drugs also, like acetazolamide and tenofovir, right? What happens in type 1 or distal renal tubular acidosis? Either this H plus ATPase is defective, or this H plus K plus ATPase is defective, or carbonic anhydrase is defective, or bicarbonate chloride exchanger is defective, right? If all these things become defective, if this H plus is not going out, this K plus is also not being reabsorbed. This K plus will be excreted out of the urine, and this will lead to our hypokalemia. Apart from that, we will also get acidosis. This excess H plus ion will combine with CaCO3, right, and form our bicarbonate, but in the process, the bones will neutralize acid, but in the process they have to excrete out calcium ions, right? And all this calcium will get filtered out into these tubules, right? But since the body is having acidosis, it is going to reabsorb lot of citrate, because citrate can be converted into 4 molecules of bicarbonate. Body will absorb lot of citrate. Now calcium has got nowhere to bind. So we got lot of free calcium. This free calcium is going to form renal stones, right? Then we have got type 3 renal tubular acidosis. It's a combination of type 1 plus type 2. In type 4 renal tubular acidosis, we have aldosterone deficiency, right? So less reabsorption of sodium and less excretion of potassium, right? So there will be hyperkalemia, right? This hyperkalemia is going to inhibit the level of glutaminase, which is going to reduce the level of NH4 plus ion, which is going to further impede excretion of H plus ion. Excess aldosterone will lead to alkalosis, and less aldosterone will lead to acidosis, right? So hopefully this video has helped you in understanding the critical concept of renal regulation of acid-base balance and conceptual understanding of renal tubular acidosis, right? Wish you all the very best. Thank you.